Good morning to one and all present here. IIIT-B welcomes Dr. V.S. Shankaranarayana Rao, Professor of Neurophysiology. He is the office in charge of Neurobiology Research Center, NIMHANS. He did his PhD and MPhil in Neurophysiology from NIMHANS and has a specialist training in electrophysiology from Wood School, USA. He has been honored by various awards like Sir C.V. Raman Young Scientist Award, INSA Young Scientist Award, Valmiki Award, Karnataka Padam Bhushan Award, to name a few. He has also worked with various Nobel laureates like Professor Susumu Tonegawa and Professor Eric Kandel. IIIT-B welcomes Professor.
Very good morning. It's a pleasure to be here with the intellectuals in and around me. Thanks to Professor Poonacha and also Professor Prasanna and also your director for inviting me to share some basic ideas about how brain works. And have you understood the brain in totality yet? And is it a scope for us to understand the brain and its modification both in healthy individuals as well as in the disease processes? So ultimately, the neurobiologists and neuroscientists, so we are looking in terms of understanding how does the brain perceives the information from the external world. How does it integrate that information? How meaningful behaviors are elicited from each one of you? Why each one of us are differentially behave at any given point of time, even though we are in the same environment? These are all fundamental questions when we talk about the normal functioning of the brain. Parallelly, you have certain set of behaviors are altered in an individual's as a result of a malfunctioning of the brain connectivity and its circuitry. So it's extremely important that we need to understand the disease process as well. It has a lot of implications for us to treat a complex brain disorders. It may be neurological or psychiatric disorders. Having said that, we have not understood the brain in totality yet. So why so? But still, the human brain produces a complex behaviors from each one of us. It is extremely important. It has a survival value too. A simple quiz for you to answer. So you have a white background. On that you have some dark patches. Can you identify an object which is familiar to you guys? Dalmatian dog. Anything else you guys see there in this picture? Cow. Okay. Tiger. Horse. What else do you see in this picture? It looks like a Dalmatian dog, but in reality there is no dog as such. There's absolutely nothing. So it looks like there is a Dalmatian dog, this is an head portion, and these are the forelimbs and somewhere like a hind limb. If I remove this portion, the head portion, even if I give 15 minutes for each one of you, you will not be able to identify a Dalmatian dog. Or someone who has not experienced how does the Dalmatian dog looks, there is no way they are going to solve this puzzle to say that, okay, it appears, this object appears like a Dalmatian dog. Never. But in reality, there is no dog as such. But your brain is programmed in such a way that the Dalmatian dog appears like this. That image is stored in the brain. Even though we provide a partial information to the brain, it puts everything together and a meaningful behavior is elicited. The image was the same. Question was the same, but each one of you had a different answers. That means the way you perceive the world is different from each other. So This is the simplest circuitry of the human brain which was involved in identifying this as a Dalmatian dog or the other species. So don't worry about it. Each one of them are the small compartments of the brain. All of them are put together, and your brain perceives the information like a visual information or an auditory information. When you saw the image and I asked the question, that information has reached into the brain. Then it processes all that information with the involvement of multiple centers of the brain. You have small regions of the brain. Each one of them are specialized to process a different sets of information. So that has been processed. Then ultimately, you said it, there is a Dalmatian dog. But what is most important is that even though you know the answer, but each one of you had a different latency period to say that or identify an object. Okay, so why so? Each one of you are so familiar with an object which is very familiar, but each one of us had a reaction times which was variable. That means each one of us were using the brain and its circuitry to solve this puzzle at a different spatio-temporal scales. Spatially and temporally, the information processing ability of each one of us was distinctly different. That indicates that individuality is a process of evolution. So why each one of us are distinctly different? No two human beings or no two species. They are alike in information processing. Thus, in a nutshell, the brain is an organ of mind. So it's a very loaded statement. We don't know what is the basis for the mind. We don't know what is the neurobiological basis for the mind and consciousness. But what we know is that a behavior and the brain. A brain and the behavior we know. So brain is a tool which is involved in processing the information available within our body and also from the external world. It perceives that information, it processes that information, and specific kind of behaviors are elicited. Beyond that, in terms of understanding a consciousness or altered consciousness is not yet understood. The neurophysiological basis is yet to be understood. Essentially, you have the different organs of our body which is fitted into the cranium, that's the head. So you have the visual information, auditory information, different kinds of sensations from the external world. They are all stimuli, sensory stimuli. And also you have the sensations within our body, reaching into the brain, processing that information and altering our behavior, and also our ability of answering.
How do you define mind as such? I mean, you have a specific definition as such?
We have certain tools, it's essentially understanding, imaging the whole brain at the different states of mind. So you ask them to do a different kinds of a complex, challenging cognitive tasks in terms of solving certain puzzles, which I showed you like this. So even while you are imaging the whole brain, these tasks can be played. Then you see that which regions of the brain are activated at any given point of time. How does this processing happens? So we feel that each one of us, we have a different levels of consciousness and the way in which we recruit the different regions of the brain to solve the task. Largely not known yet. Where is the seat of consciousness is yet to be understood. What is most important is that in the beginning I said largely the organization of the brain itself is not completely understood. So probably because of three important reasons of the complex organization of the brain. One is in terms of its numbers. You have a trillion nerve cells, trillion neurons are loaded in our brain. Then you have the speed in which it processes the information. So it ranges somewhere between 0.25 milliseconds to 1 millisecond is the speed in which one nerve cell to the other nerve cell to communicate. or I have a command which is originated in the brain, from there to reach into the spinal cord, it is about a meter, it takes about 0.5 millisecond or so. Then it's quite fast, the communication. So it ranges somewhere between 10 to 100 meters per second. That is the speed in which the information is being conveyed from one cell to the other cell, or one region of the brain to the other region of the brain. And most importantly, so any given cell receives multiple inputs of multiple origin. The circuit which I showed you, you have a single cell which receives the information from the multiple sources. It ranges somewhere between 10,000 inputs to 2 lakh inputs onto any given neuron. Then you have the complex organization is the network. The brain's connectivity seems to be a lot more complex than the cosmos itself. So it's something like, an analogy is 15 times the radius of the earth is the connectivity what we have in the brain. So is it because of the complex organization of the brain, either in terms of the numbers or in the speed in which it processes the information and its connectivity, does this limit the biologists and the neuroscientists to understand the brain in totality at any given point of time?
Sir, I'll say something for us poor computer scientists. The volume of the microelectronic circuits, the surface area is only about 1 square centimeter, and 100 billion of them will be about 1 micron. So on a volume basis, it's much more efficient, but no way it comes anywhere close to this capacity.
So
we build small things, but they are down.
Yeah. So so essentially you have several thousands of microchips are embedded, and which is dynamically altered on the environment. So it's all dynamic. The brain's connectivity is dynamic. That's what I am going to discuss today. How dynamic is our brain? How soon we could modify those complex brain circuitries. So that is helpful for us to induce the reaction times for us based on the requirement. Having understood such degree of complexity, in fact, the brain is the first organ to develop. If you look at the development of the brain, the primitive brain is very much there by 25th day of conception. So immediately after conception, by 14 days, a primitive brain development happens. The brain is the first one to develop and to mature to significant effect before the child is born. If a child is about 3 kilos, you have 900 grams is the weight of the brain. The roadmaps has been laid down, its connectivity has been established, but the modification and altering the circuitry by experience is going to happen till our death. So since it develops much early, look at this is at the age of 1 year. So this is in this portion on the surface is called as a cortex. Don't worry about these complex anatomical names. And look at here. So this portion of a cortex, you have these cells, each one of them, they have the processes. These are the processes which communicate to each other. And this is from somewhere here. So different regions of the brain, their maturation and the connectivity varies. So some of them are very early mature, and some of them they take over a period of time up to the age of 2 years. Up to 2 years, so we call it as a critical period for the development of the brain. So brain is highly plastic at that time. It can be modified and modulated in terms of its connectivity very robustly in a rapid scale for both good as well as bad. But that plasticity continues up to the age of 20 years. So look at here, the number of cells have not significantly altered, but its connectivity has been enhanced very large scale. Can you compare this with this and this versus this? Look at this, 1 year to 6 years. So its connections have significantly increased. So what that mean? the circuitries of each one of these nerve cells and its ability to process a particular set of information that determine whether to regain such a complex connectivity or to remove it. So all the time the removal happens. It's a phenomena in the nervous system called as pruning. So you have a connection. No longer I have the communication between these two guys. Take out this guy, the new guy will come and occupy. No space is wasted at any given point of time. The connectivity is extremely important. You have a wonderful, efficient connectivity within the brain system. Look at here. So it's essentially simple terms. Okay, no, don't get into the technical aspects of it. So here is a 4-year-old child brain which is mapped in terms of its maturation based on the activity on the surface of the cortex. So this is on the surface. So you have the red one, the colors, which is less matured, and this one more towards the blue, he is more matured. So this is at the age of 4 years. If you notice here, a 4-year child can make a discretion between different kinds of stimuli, somatic sensations. It may be pain, or it may be touch, or it may be hot or a cold, each one of those sensations. Discrimination happens very, very significantly, and that remains forever. So every time we are not going to experience, okay, to touch a candle and a glowing candle and see that whether it will burn, whether it generates the heat or not. This is a single time experience. The maturation happens much, much before. And also the moment the child is born, you have the visual information. It looks at the environment, and the brain matures as and when the child is exposed to the different environment. So you have this portion, the visual one, and the sensory information processing. The maturation happens much before, because when the child is crawling, it could perceive the touch, pain, and each one of it. Then subsequently, the intellectual ability of an individual happens up to the age of 20 years. The juvenile law, which want to be modified by the Verma's commission, they said that it should be reduced from 18 years to 16 years. And we had a delegation, then we represented the neurobiology group, and then we have to educate the judiciary that why these guys are not able to discriminate between good and bad. The reason is that the maturation and decision-making regions, this portion of the brain, happens up to the age of 20 in humans. It's not the reproductive age. The reproductive age is reduced, but the brain maturation and making the judgments, a logical thinking, individual judgments, continued up to the age of 18 to 20 years. So that means essentially you are talking about the behavior of an individual is do's and don'ts to the environment, which is strongly guided by the brain and its circuitry. On the other hand, you want to drive with the instincts. Okay, all the rapes and the sexual acts which is happening prematurely, so that's all driven by the instincts. So one is on an instinct, another one is on to the commands which are originated from the brain. So don't you think that we need to balance both? Look in terms of a scientific perspective and see at what is the best age to punish these guys.
So
okay.
When you say instinct, is it not a brain activity on itself?
It is a brain activity. It's very much brain activity. So over a period of time, based on the environment, so you have most of the reproductive age in both male and female is coming down. So it's a kind of exposure. And you have certain regions of the brain, so they process this, the reproductive and the sexual acts. It's completely driven by the brain. So you have the specific hormones and the certain regions of the brain, and they determine the reproductive cycles. It also determines your sexual acts.
Is there any threshold where the behavior based upon instincts get lesser proportion compared to, for example, behavior based upon instincts?
So I am going to give one example subsequently. So you have evolutionarily we are like, our cousins are monkeys. What is the distinction between a monkey and a human in terms of a behavior? Do's and don'ts. We know exactly, even though instinctively I want to react to someone, and we look at the environment, our behaviors are determined by the environment. So there is a do's and don'ts. So why so? This portion is called as a prefrontal cortex, is largely evolutionarily evolved in human species. So this portion, the medial prefrontal cortical region, which is responsible for the judgments and a logical thinking and a complex acts what we perform and a skilled acts, each one of the skilled acts, because we have these fingers. So that is evolved by this one. But in the lower species you don't have. But in certain psychiatric illnesses, the problem is that the inputs from this region to another region which is in the middle of the brain, all the time it drives. Perfect, no problem, please go ahead. But this guy will say, no, this is a classroom, don't do that. So you are in the classroom, so you cannot do this one. Don't speak loudly. So she is your colleague, you cannot behave like that. So the comments are coming all the time. The do's and don'ts are determined very significantly. But in some cases, it so happens that they are not able to distinctly differentiate. Yeah, I think it is working, yeah. So the problem is with the cortex. This is called as a character cortex. It's exactly situated here, okay? Your characters are determined by the information processing from this region. Now you have another region which is in the middle of the brain. It's called as an amygdala. So they are all older ones. So they will say that perfectly fine, jump now. You fight with them. You behave rudely. You react. Irrespective of the place, irrespective of the position, irrespective of the environment in which you are at this point of time.
So it's animal behavior versus...
We are all, we have all species brain is there inside. In fact, so you have all species brain is there. So on top of that, this is the larger one. The cerebral cortex is evolutionarily evolved in a bigger scale in the humans.
So I just want to go back to what you had on your previous slide, that experience contributes to the development of the brain. How do you juxtapose that statement with something like this, where you say by the age of 20, the development?
Or even is the intelligence, logical thinking, complex mathematical calculations what we do, all the skilled acts what we perform, you gain by experience over a period of time, the maturation. But the maturation and the logical thinking is determined by the environment. So it's not necessarily each one of us should have the same judgment, and also we should agree for the same point. It will never happen. That's the beauty of the brain. So because of a heterogeneity, then if you have 10 people in the committee, if 6 people are agreeing, you consider that is reasonably good. So 4 people, they might have their very strong views. So it is a permutation and a combination with the algebraic sum which you are going to use to decide the decision of a committee. The same thing happens in the brain as well. So you have both kinds of inputs, the do's and don'ts, and also the decisions which you are going to make, and most importantly, decision making. What is that translates? My rude behavior will translate into something in the classroom. What is that it's going to translate? We know. If I don't perform well in this semester and overall score decreases, and what is its impact onto your career? You see that one? You need to have a conscious decision making which happens all the time for each one of our act, and that has a survival value. Each one of us, we survive based on that. Thanks to this one. So highly evolutionarily in the human species, this region of the cortex is highly evolved. So that will determine your characters, the do's and don'ts. And also, that suppresses the activity of the older cortical structures and also the subcortical centers which are sitting in the middle of the brain. So they are just waiting to come out. That is the reason while you are in a deep sleep, many things what we experience which is not true, which is not in a realistic world. Because this one, the neocortex, the newly evolved cortex, what we have in the human brain, its activity goes down. The middle of the brain, its activity comes up while you are in the deep sleep. So that determines the balance. That's how we know that, okay, the neocortex is all the time checks the signals and the commands which are originated from the older cortical centers or the other regions of the brain as well. What is very interesting is that, don't worry about these names. This is, if you look at the surface of the human brain, it looks something like this. This is a schematic. Okay. So this is the front of the brain, this one, and this is on the back. So all the visual information processing happens here, and you have the higher order cortical functions, including judgments and logical thinking happens here. And then you have certain small regions of the cerebral cortex. They are able to process very defined information related to the different kinds of sensations or the information. Say for example, smell, the auditory one you have here, you have the olfaction, you have the taste, and then you have the speech generating one which happens from here. Okay.
But I believe that the brain has a unique ability of one area taking over the functions of another in case there is some damage and so on.
That's what I am going to talk subsequently.
Okay.
That is the plasticity, how much it can take over. So what is the magnitude in which it could correct by itself? It's called as a reorganization of the brain whenever there is a damage occurs. It happens by itself. So each one of them seems to be highly specialized to process a particular set of information, then integrate all of that. They are called as associational cortical areas. That means your visual information, auditory information, you touch something, everything is put together. But our behaviors is based on the multiple sensations. The multiple stimuli is being put together, and you have the integration which happens in the brain. How does this happen? Okay. So it's essentially each one of these regions are evolutionarily evolved to process a particular set of information, maybe because they are structurally different, distinct. Each one of the regions, small compartments, structurally they look different. Essentially each one of them are the nerve cells, and they are distinctly different. Structurally they are different, functionally they are different, biochemically they are different. The kind of signals which are generated from each one of them are different. The kind of the biological chemicals which is being synthesized and processed, they are distinctly different. So say for example you have a cell something like this. This is the one which is controlling my equilibrium and the balance. So your body in a space is sensed by this kind of a cells which is present on the back. It's in the cerebellum. And you have a cell something like this, okay, that's in the cortex. It's giving a commands for me to move around, the motor controls. And I have a cell something like this, which is there in the middle of the brain, a structure called as hippocampus, which is crucially involved in learning and memory. You have this kind of cells. Something which is responsible for the carrying the information from the periphery in the middle of the brain. You have the cells something like this. Thanks to the nature for generating a multiple brains within the brain. So each one of them are extremely complex and distinctly they are different. How do you know that they are distinctly different and they process the different sets of information in an orderly fashion and generates the behavior? Look at here, somewhere here, this is the cortex. You have a nerve cell which generates electrical activity something like this. They are the action potentials, something like this. It just fires for some time, then it sleeps. Here is a cell which is present here. They have a burst activity. It fires very rapidly, then it just rests for some time. This is a cell which I was talking about, which maintains about equilibrium and balance. It fires something like this. It just fires, rest, again fires, rest. Something like this. Or you can also have very periodic the electrical activity is being generated, something like this. The rhythms. So you have a very particular pattern of generation of electrical signals in the brain, and each one of them are extremely important for information processing which happens. Or here is an example, same cell, same nerve cell can fire at different frequencies at the different point of time. Say for example, a thalamus which receives all kind of sensations before it sends into the cortex. You have the cells, they work like this, very regular firing, something like this. You have very rapid firing, or you have the burst firing, something like this. When a cell is like this, initial attention, when you are very seriously processing the information and working, it can get into this phase. By evening it can get into this phase so that you can sleep.
So it's a variable clock rate.
Same cell, same nerve cell, based on the requirement of an individual, it fires at different patterns of electrical signals, and these electrical signals are the signatures for communication between one cell to the other cell. Look at here, these guys. So you have a particular set of clocks of electrical information processing in the brain, or the signaling in the brain.
So are all the cells roughly firing at the same rate at a given time, or different?
Oh no, they are distinctly different. Each guy is a different time. Okay, because each one of them, even though they are localized in the same region, they have to conserve the energy because the strength of stimulus is different. Each one of our brain, at any given point of time, how many cells are recruited to process that information is distinctly different. So we are talking in terms of thousands and thousands of cells in a given region of the brain. So they are distinctly different. So some is in a phase mode, some is in a tonic mode. So they go in cycles. That's how the biological rhythms and our moods are decided from morning till evening. Okay.
And I can see that on this picture, the noise at 50 millivolts is very small. There's no noise at all. Whereas electronic circuits, will not work at anything like 50 millivolts. I think Punaka can tell us more. We are minimum at 700 millivolts or so.
So these are intracellular recordings. You put an electrode inside the cell body of a neuron and you are recording it. But if you are recording from the extracellular one, the noise is extremely huge. The extracellular matrix, the connectivity what you have, so you have the source and sink problem comes into it. When we record the EEG, that's a big issue for us. That's why we have to go to the better technology like magnetoencephalography, so you reduce the noise to the best of your ability.
But the underlying physical process is so noise free.
If you do it intracellular, something like this, inside.
Okay, and even a synapse, across the synapse?
Across the synapse, I will show you, because it is very, very noisy. Okay. So when we do the extracellular recordings, that's outside the cell, you have the electrical communications are happening. When you record it, it has a significant noise. The EPSPs, it may be excitatory postsynaptic potentials, the communications, or it may be the inhibitory signals or excitatory signals. So here, inside the cell, you put an electrode, you record it and then amplify. That's what you are going to visualize it here. These are very, very clean. But in the nervous system, in reality, when they are talking together in a network or in a circuit, they are not clean like this. In any region, it's not clean. So even in the spinal cord, it's not clean. So essentially, if you look at a single neuron, it has a multiple components. This is the cell body. So you have the processes which is originating, they are called as the dendrites. They act like an antenna which receives the information from the multiple source. You have a small button-like structures, they are called as the dendritic spines. And these are the ones which is responsible for receiving the information from multiple source. And these are the sites where the communication happens. So the junctions, they are called as the synapses. So communication between one cell to the other cell, where the communication happens, they are called as synapses. And where does this happen? So this happens onto this dendritic tree, which receives the information from the multiple source, something like an antenna. It receives the information, and then it sends that information to electrically to the cell body, where the integration happens. So what do you mean by integration? So you have some inputs, they are excitatory in nature, so they can excite this neuron, and some of the inputs which can inhibit, it can suppress the electrical activity of a particular neuron. So it's essentially how many excitatory and how many inhibitory inputs are there, both put together, you integrate it. Then you have the electrical signals which are originated here, they are called as the action potentials. They are the one, they carry this information which is being processed by this cell and communicates to the other cell. So by these cables, they are called as the axons. So you have the integration happens here, the electrical signals are carried to this cell, they come and communicate. Receive the information, integrate the information, and encode them into the electrical signals. They are the action potentials, and they reach into the terminal where the communication happens. All throughout the nervous system, whether it's a unicellular one or the humans, this is what its role. The nerve cells, they have this particular role. Receiving the information, integrating the information, processing the information, communicating that information which is being processed. That's all its function. Either you take an amoeba where there is no nervous system as such, or you take a cockroach, or you take the chimps, or you take the humans. All nerve cells, they are designed to process this information.
So where do the decisions happen? So how, so if there is a processing, so the emergence of decision, how is it seen?
It is a multiple brain regions. They have the information is put together. So the decisions is not by a single neuron. The decisions are made based on the previous experience where the memories are stored related to that particular aspect. Now you are acquiring the new information, both are put together. Given point of time, how attentive you are and what is the relevancy of that information which you have learnt, proportionately your responses are originated. The decisions are originated. And again, the decisions for different kinds of our behavior are originated in the different regions of the brain. So it is not in the same, there is nothing like the region which is involved in all kinds of behaviors what we perform. If it is related to my motor acts, it's different region. Related to your language, it's different. Related to your intellectual ability in terms of decision making, they are all different. But all of them are integrated between both halves of the brain, both hemispheres. They have to talk each other. They talk each other before the decisions are originated.
So when we tell the regions speak, so what do we mean? I mean, do the cells in those regions speak to each other, or is there something?
Oh, they speak. In fact, that is the reason I told you that you have a very, very extremely complex network. It's about 10 to the power of 8 meters. So imagine that what is the amount of connectivity what we have. Even though the brain weighs about 2% of our body weight, it's about 1,400 grams. But imagine the connectivity. And also, nothing is processed with free of cost. The energy. 30% of the nutrients and the blood in our body flows to the brain. So this clearly indicates that the information processing is an active phenomena. Nothing happens freely. It's always with the expense of energy. So you have about 30% of it. So even though the weight of the brain is about 2%, but you need 30% of the energy to the brain. So essentially, it's important to note that the communications between one neuron to the other neuron, or the multiple neurons and one region of the brain to the other region of the brain, everything put together, that determines your intelligence. So intelligence is the overall activity of the brain with the multiple centers or the brain regions. They process the information It's a complex behavior which is elicited based on the experience. It's a complex one. So the communications between the neurons, they determine this one. And most importantly, it also determines the brain's ability to generate the complex behaviors. So many of the behaviors of the human species seems to be a lot more complex compared to other species in the world. Why so? Probably the requirements are different, and the organization of the brain and the information processing ability of the human brain seems to be distinctly different compared to the other species, because there is no requirement for them. So their requirement is entirely different. The lower species, like our cousins, if you look at the monkeys or chimpanzees or baboons, their requirements are entirely different. But each one of our requirements are entirely different based on whatever the professions you take. Accordingly, certain regions of the brain is more used, and certain regions of the brain is less used.
I think that theory, it's a self-feeding, because did the requirements come from brain, or because of the requirement the brain is like that, is a question which...
First of all, you have to perceive what is the requirement, because who is going to perceive that? So what is the implication of attending this lecture is determined by whom? So even though your professor has forced you to attend my lecture, but there is an issue, okay, this is what is the content, probably it is going to be helpful for you, but your professor can drive you inside the lecture hall, but your brain is not tuned by your professor, but you have to tune by yourself, whether you are motivated to listen to my lecture. And if it is not relevant for you, even though you are sitting very much here, and we feel that, okay, you are so attentive because you are looking at us and your eyes are wide open, and your gestures and postures predict that you are very, very attentive. But that is not true, because in the brain you are processing something else at this point of time. At 5 o'clock you have something need to be organized in the lab, or you have to submit a report to your director. That is more important at this point of time rather than getting into the details of my lecture. It's very, very true. All of us, we undergo. All normal humans, this is very, very common. There's nothing abnormal about it. Don't worry about that. But the point which I want to make is that the degree of attention and the reward what you are going to get, whether you are motivated to learn that, it has any relevance for you. Something which is not relevant for you, there is no way that the brain will waste its energy to process that information and translate that learned information into memory. It's a very active process. The brain don't want to waste its energy for that purpose, because you are using that energy for thinking about how the report has to be prepared, and is it possible for you to submit by 5 o'clock or not. Or you use an excuse that you attended a lecture, my lecture, so 1.5 hours has been wasted, so I am not able to submit the report by 5 o'clock, tomorrow morning 10 o'clock is fine. You understand, that's where the brain, the decision making and also the alternatives are originated at every point of time. How does that happen? Look at this guy. A single neuron can talk to the multiple neurons with different degree of communication. Some are very strong communication, some are weak communications. So even though the information which is being processed by a single neuron, it carries the information, it communicates to the multiple nerve cells, and each one of them, they elicit a different response, because the perception of that processed information and the chemistry which is involved in communicating the information will be distinctly different, whether you perceive that information or not. So you have the different kinds of receptors which they sense the chemicals which is being released by these axons, something like this. So depending on how much interest you have showed in my lecture, accordingly that much quanta, that much amount of chemicals are being released over here. That much amount of chemical which is being available here, that activates this neuron. it communicates that information effectively. Some is for a brief period of time, it's perfectly fine. So some of them you feel that, okay, from the point of examination or from the point of your project, it's extremely important. What you will do, there's a sustained activation, it happens. So I'm going to tell you, how does these memories are formed and what is the relevance of that? Completely based on this one, the connections. And most important thing about the brain is that its unique ability to modify based on the requirement all the time. So brain is capable of undergoing extensive reorganization, remodeling of its circuitry and its connections for both internal as well as external environment. So from the body, different parts of our body, the signals are reaching into the brain and the processing happens. And then you also have the environment which is affecting, and also you have the biological changes associated with the different disease process. All of them, they work together. It can induce an alteration in the circuitry which I was talking to you. The brain's ability to process the information can significantly alter. So this phenomena is called as the plasticity. Brain is the only organ which is highly plastic in nature. Imagine if brain was static in nature. Whatever we have learnt in the 5th standard, that remains forever. No scope for me to understand subsequently. Then what will be the fate? So thanks to the nature has generated this complex brain with a modifiability and a flexibility, with a greater plasticity, which is not there in other regions of the body. In the other regions of the body, other organs of the body, you don't find that. So the brain is the only organ which has a greater plasticity. When I say that it has a greater plasticity, how do we understand? What is our measure? The measure is something like a behavior. So every time some changes in the circuitry happen, your behavior alters. So that one could understand how a particular behavior is generated, that happens with the involvement of multiple centers of the brain. And each one of those centers are specialized to process a particular set of information, which I showed you. Visual information is being processed specifically from a particular region of the cortex. And each one of these nerve cells, they are specialized to process a kind of information, because structurally, functionally, and biochemically they are distinctly different. Then each one of these nerve cells, they talk to each other, so you have a complex microcircuitry, something like this. So you have a whole lot of biochemistry. If you look at the number of genes which are expressed in the nervous system, is greater than any other organ of our body. So that clearly indicates that a specific genes are expressed in the different regions of the brain and different cells of the brain, which I showed you. Maybe this heterogeneity drives a lot of the protein synthesis and the gene pool. If a specific gene is altered, it could also alter the behavior. So essentially the neurobiologists, we use this as a template. A cognitive biologist, they look into the behavior. A systems biologist, we look at this one. Electrophysiologists, they look into these connections, and the structural biologists, we look into the structural connectivity. And then you have the biochemists, you have the geneticists, whatnot. Everyone contributes for the understanding of the brain, depending on the expertise which they have. On top of this, you have the information technology people like you guys, where you are going to integrate everything. Put everything as a package and you say that this is what is happening. You predict it. So even though many times it doesn't happen in a realistic sense, that what is the prediction, I can do it from your brain. What is the behavior which is elicited? But to some extent, you can say that a particular framework of a rules does followed by each one of us, which I showed you that each neurons, their ability to process the information happens very uniquely in all regions of the brain, irrespective of the type of cells. So it's extremely important that how we are going to study these plasticity mechanisms. The most important question is that, is all the regions of the brain, they are equally plastic? So imagine all the regions of the brain is so plastic. So every day I walk in a different style. Does it happen? No. There is no requirement for me to walk in a different style. So our gestures and postures, they are the signatures for each one of us. Okay. But certain regions of the brain, they need to be extremely plastic in nature. So every time you are learning new things, so you have the structure which is embedded here, which is demarcated in the yellow color, is called as the hippocampus. This is the structure which is crucially involved in learning as well as memory. So whatever you are learning, the activity in this region determines whether how long you store that information. So is it till the end of my lecture, or till you leave this building, or it remains forever? So that is determined by what is the kind of activity which we have in the hippocampus. So this structure seems to be highly plastic in nature, thanks to the nature, because this structure is so plastic. That is the reason every moment you could learn new things, and if it is relevant for you, then it translates from short-term memories to the long-term memories. If it is not relevant for you, for a while you remember that, after that you don't remember it. Okay. So essentially you have a complex behavior is the learning as well as memory. It's a higher cognitive function of a human species is the learning as well as memory. So in a simple terms, learning is the acquisition of new information. Anything you acquire new is a learning process. And whatever you learned, that information, if it is relevant for you, you are going to store it. So it's called as the memory. The retention of newly learned information is the memory. But your memories are keep on altered based on the experience. So from one class to the other class, one semester to the other semester. So what happens? You have that information, what you learned in the previous lecture. On top of that, something else will ride. So that is called as consolidation of the memory. So essentially, you are refining the memories, whatever you have learned. And then reconsolidation of the memory. That's the most important one. So if it is relevant for you, you are going to revisit the same thing again and again. So consolidation and reconsolidation of the memory which happens. These four aspects which determine your intelligence as well. So what you acquired, how long you stored, and how quick you retrieved it. So many times it so happens, after the examination, if you ask me to answer all the questions, probably our scores will be much higher, because you have already activated those memory circuitries. So after the examination, there is no pressure, no. So if you solve the same thing, you will solve it much better, because the memory circuits are already activated. So essentially, you have the memories, some of them for a fractions of seconds to seconds, and they are called as the immediate memories. Some of them are the short-term memories, that is seconds to minutes, and some of them are intermediate, that's minutes to hour, and some for days to years, permanent memories. So memories have a temporal scale. Who decides actually what need to be stored for how long? Who is going to decide? What is the basis for that? How does your brain process that information and say that this is relevant for you, please store it? How does that happen? With the involvement of multiple regions of the brain. So different kinds of memory, different regions of the brain, they are primarily involved. So it's not that only one region is involved, that is primarily involved in processing that information, but integration of the information happens across different regions of the brain, and the memories are stored. Okay. Let's not get into the technical details. Okay, I will also not give the examples of humans. One example I will give you.
One question is,
yeah.
You still, so that diagram I was looking at, so the relevance of the model like semantic and episodic memory is still very valid, is it?
Oh, it's very valid, because essentially, we call it in a different names. It doesn't matter. So it's kind of a explicit memory or an implicit memory. So if I am a psychologist, I use a declarative memory, non-declarative memory. So if I am a neurophysiologist, I will go for, looking in terms of, the explicit memory or an implicit memory based on, it's all the same. The way we perceive that information is in a different context. That's all. So otherwise, they are the same. So broadly, they are like a declarative one. A conscious participation of an individual is required to acquire that information is called as a declarative kind of memory. It's a pool of memory, but it's beyond that. And non-declarative one, many things it happens even though you are consciously not participating. Just like climbing a stairs, riding a bicycle. You are not going to consciously participate in that. Once you learn, then those memories are there. And consciously, you need not learn every time when you climb the stairs.
But something which is explicit can be made as implicit.
They work together. So there should be a reason for you to climb stairs, no? So who determines that? The hippocampus will say that, yes, your boss is called, immediately you report. So you will see the relevancy of that. You are going to learn something, or you are going to get good or bad, whatever it is. So there is some kind of a motivation and a reward which is associated for you to perform these non-declarative tasks. So both of them, they work together. But you have a neurobiological sense, they are distinctly different in terms of its ability to process the information. But our behaviors, you have both of them together. They have to work together, the behaviors.
Just a general question. The simplest memory which computer designers know is 0 and 1. Now what, how, and it is asti nasti, it is basically existence, non-existence.
Yes.
What in the brain's memory refers to existence, non-existence, or is different portions?
Brain will read only two things. For its everything is the kind of electrical signals and the communication. So it doesn't know what is the relevancy of that. One is in terms of the strength of the stimulus and the duration of the stimulus and the frequency. Like your electrical circuits, what you have, the same thing happens here. How long it has been activated, the connections which I was talking, how long it has been activated, how strong was the communication, and what was the strength of the stimuli, that determines how many synapses need to be recruited. So you have very small input which is reaching into the brain. I have very little neurotransmitter is released. It could activate only 5 synapses, for example. I am so keen to this. This is so fascinating. The brain is so fascinating. So you put a lot of effort for you to make a notes and see that how best you could put all your efforts to understand it, and then you have lot more activation of this particular circuitry. And then after the lecture, you go back and you refer something. It's a recapitulation of the events, what I said, whether it was true or false. That's where the consolidation happens. So it need not be necessary to activate the initial pathways, but subsequently from hippocampus it translates into the cortex, the consolidation. And also reconsolidation happens while we are in a deep sleep. The memories, short-term memories to the long-term memories, and the relevancy doesn't happen now. It happens only while you are in a deep sleep.
So can dreams be one of the generations of that kind of reconsolidation as well?
You see, there is a sleep stage called as the rapid eye movement. During that phase, a small portion, about 5 to 10%, may be the dreams. So 90% is used for storing the memories and consolidating the memories based on the events from morning till evening, and the relevancy, everything is put together. It's a replay of events from morning till even it happens without your conscious participation, because those circuitries are lot more active. While you are sleeping, your brain uses more energy than what I am using now. Because more regions of the brain, more connections and more circuitries are consulted for the formation of memories, for the storage of memories.
Is there it is easy to remember shlokas which are rhythmic versus just prose. Is there something, so have you identified what in the brain will enable us to store shlokas in a natural fashion versus just text? Whereas for a computer, the two are not really different. Whereas for a human being, rhythm and chandas, etc. is very important.
So that's what I was mentioning to you, that so none of our behaviors are unimodal in nature. They are all polymodal. So that means visual, auditory information, you have the olfactory and tactile. Many things. Multimodal are the polymodal information processing and the integration which happens. So all the time, this declarative kind of memories. So what are those? Essentially the facts and events, places and faces, and the spatial navigations and the spatial maps. All of that is the declarative memories where the hippocampus is involved. It is always a context base. Even though you have all these contents in the neurobiology books, but why you don't learn and remember all the contents? Contents of the books are not remembered to the best of ability, but the hippocampus one, it's a repetitive. You have to put a lot of effort for it in a context base. What is a context? You will remember about my face, you will remember about the postures and gestures of mine, and also you will remember about the context in which this lecture you listened to. So the context with respect to a particular background, the new information is being written. Okay, so you are learning with a teacher and without a teacher. So how does this happen? So most of this learning, the declarative kind of memories, the facts and events, what is there in the textbooks and the lectures, faces and places. So we learn a lot more things when you associate with so and so. The memories are formed very robustly. So that happens with the declarative kind of memories. So there are other kind of memory. They're called as the emotional memories. It's a single shot learning. You are just passing through, an accident happens in front of your eyes. There is no replay of events. Very quick memories are formed, and that remains forever, like a PTSDs, post-traumatic stress disorders. Some event occurs which is so robust, but that remains forever. So there is a different regions of the brain which is involved. This is called as the amygdala. Something learning which is associated with an emotional component. This region is involved. So different regions of the brain, they process a different sets of information related to learning as well as memory. Each one of them are distinctly different. That is the reason you have different regions of the brain. They participate in both learning as well as a memory. It's a lot more complex than what I am speaking today. How do you know this? This region is involved in this particular component of a learning or a memory. How do you know it? Any guesses? How do you know in a human? Yes, he is extremely intelligent and his memories are very, very robust and very strong. But my memories are quite poor. How do you know that?
Responses in that particular area.
So imagine that, okay, how long you store that information, and then if I don't retrieve that information and you feel that, okay, I cannot store the memory. Okay. So it does happen in the disease conditions.
It might be a problem with just retrieval.
Okay. So you have the different phases which I was talking to you. One is in terms of acquiring. So learning itself is so poor. It's not causing a robust changes in the circuitry. So thereby strong memories are not formed. If strong memories are there, very much there, I was answering perfectly fine, 15 days back before I had some problem in my brain where the blood circulation is affected. Then after that I noticed that my memories are terribly poor. So it does happen in certain disorders or due to the insults. So that's how we know that the different components of the memory related to our behaviors are associated with different regions of the brain. If that particular region of the brain is affected by a disease process, that particular component of a behavior or a memory is affected. So you have some, we call it as a reference memory, working memory. It's like a day-to-day activity. I should know actually how to reach my office, how to organize myself. They're all working memory. Or suddenly some visitor comes and you should be able to organize a chair for him and you sit and discuss. You don't plan anything, no. We don't plan what I need to discuss with so and so. It's only for the examination we plan it. But rest of our lives there is no plan. Many things it happens without planning. That means, you know, you need to have this capability of the working memory. So some individuals, it doesn't happen. So there are multiple components of memory. So let us not get into the greater details of that. One patient who contributed maximum for understanding the neurobiology of the memory, his name is HM. Okay. So this patient, at the age of 10, he started developing the seizures, the fits. A common term is the fits, seizures. So the patient, you know, this patient was treated with the drugs to reduce the abnormal electrical activity which was responsible for having the seizures, epilepsy. Irrespective of the combination of the anti-convulsant drugs or anti-epileptic drugs which was available in the market at that time, so all of that has been tested, his seizure frequency has not decreased. So say for example in an hour, 10 times he had the seizures, a generalized seizures, and he will reach into an unconscious state and he comes back to the conscious state. By the time he could recover, again he had another episode of the abnormal electrical activity which translates into the seizures. Then they identified that the hippocampus, which I mentioned to you, and neighboring regions was the site for the generation of abnormal electrical activity. They performed the surgery when he was at the age of 27 years. They removed these portions. So this is where the amygdala, where the emotional memories, and this is the hippocampus, and these are the neighboring regions. So they removed it. Both sides performed the surgery, they removed it. Hippocampus and neighboring regions are removed. His seizures have completely halted after the surgery. No longer he had any seizure episodes because the site for the generation of abnormal electrical activity has been removed. The foci of the seizures has been removed. What is most astonishing is that before surgery, whatever the memories were stored, it was intact and he could retrieve it back. But he was unable to form the declarative kind of memories, to remember about the facts and events, to remember about the faces, to remember about the places. Absolutely, till his death, it never came back, in spite of several rehabilitative protocols and the counseling protocols and the cognitive therapy protocols has been executed onto this particular patient. One single patient who contributed maximum for understanding the cognition, including learning and memory, is only this patient. What is interesting is that before surgery, perfectly fine, all memories are intact. After surgery, no new memories were formed in this individual. So every time the doctor has to go and introduce, I am so and so. He cannot even remember the name. And every time the caregiver has to show him, this is the restroom, this way you have to travel. It's in the small room. Every time you have to. No space. The spatial orientation has been lost. And you are not able to identify any of the individuals with whom you are associated, including his parents or his caregivers or his doctor. So imagine that. Don't you think this is a very, very severe impairment? You are socially disconnected.
But he was able to continue walking.
Everything is perfect except this component, even including his, most of the procedural memories, like a non-declarative one, associative memories like a drawing, writing, all this is perfectly fine. Only this aspect was lost.
But he had language comprehension.
Yeah, language comprehension was very much there. So you will remember but it is unable to store that information. The language is perfectly fine, he will communicate, but no memories. Yesterday what he spoke, and even what he had at 10 o'clock for his breakfast, he is not able to even to say that what he had. You don't remember what he had at, when if you ask him at 12 o'clock.
So the formation stopped, or even the earlier memory?
So essentially, what was the problem with this guy is that first is even though he used to acquire the information, it's for a very brief period of time. It was never translated into long-term memories. Translating short-term memories to the long-term memories is the function of hippocampus and neighboring regions. Since he doesn't have the hippocampus, he is unable to translate that information. Since he is not translating it, there is no storage question comes, there is no recall issue comes. It was very true. And he is the one. So he died at the age of 82. And this patient has completely opened up the area of cognitive biology. The one patient, his study has opened up the area of cognitive biology, and the neurobiological basis of learning and memory has been studied in great detail. Okay. So most of the time, a disease process with the symptomology and the abnormalities that helps us to design the experiments and perform in the animals, and then you study it.
Was he able to do, let us say, give image recognition and discriminate new images from old images? Was he able to do this?
Emotional memories were perfectly, to some extent it was fine. So if you flash the picture of his mother, he can perfectly identify, because the memories were there before surgery. But the new things, like including his own a physician or a doctor, if you flash his picture, oh, I don't remember who is he, because he has been treating from last 20 years.
But could he distinguish the new one from mother?
The mother only he can identify, not others. That's why this was a very, very robust case where the places as well as the faces, which is the function of the hippocampus recognition, was lost in this patient. It's a clean case till his death. It was never improved at all. It's more like an all or none. Switch has been turned on and off. So before surgery he was on, after surgery he was off. Only to this component of it, only the declarative kind of memories.
He was walking,
everything is perfect. Everything, rest all is perfect. In fact, a lot of motor skills were given, because that is a tool to improve his memories and things like that. But not this one has not happened.
You could drive and so on.
Not really, because you don't remember the maps. There is no spatial map, because he is not able to identify where is the restroom.
Okay.
No map is formed. No spatial map is formed. So it's very, very severe. It's a very, very clean one. Then this patient has provided a lot of information about understanding the biology of the learning as well as the memory formation. Then in 1960s to 1970s, the scientists and the experimental psychologists and neurobiologists, they started mimicking the different disease process which is reported in the different places. Different diseases associated with the brain. So if you lesion this one, you remove small portion of the brain, whether it translates into any behavioral abnormality, what component of memory is damaged, and things like that. A lot of experiments were designed to understand the role of hippocampus. So one such behavior which is here, something like this. So this is a tool to assess the spatial map. So next time when I visit, I should be able to come to this place without having any issue if my hippocampus is intact. So otherwise I won't be able to form these spatial maps. So what you have is, you have a pool of water here, there is some hidden platform here, and there is a small checkerboard there which is hanging. So when you enter, everything looks alike, only in one place there is a red board and it says that exit. So that's a cue for me to leave this hall. If I have to leave the hall, this is the cue. So it's a spatial map. So each one of us, we have these spatial maps are formed. Now, if a hippocampus is damaged, then what happens? So I will not go to the videos. I'll just show you this one. Now imagine that I did a surgery, remove a small portion of hippocampus, and if it is damaged both sides, this is what happens. So this is the one, the normal one. If you just train these guys for a couple of days, he knows that, okay, every time it has to swim in this direction so that it can escape from swimming, because by using this cue. So it will not swim in this direction or this direction, it moves only in this direction. So there is a spatial map, and there is an association between the different objects which is there, and then you move in that particular direction. Now if hippocampus is damaged, this is what happens. So you are not able to identify a place. So the external space is mapped in the hippocampus. So those cells, if they are abnormal, then you are not able to have a spatial map of your external world. So the hippocampus is extremely crucial. So that damage has happened. Then in 1970 to 1980, that was the time. So thanks to the physicists, they started developing amplifiers and all those tools, powerful tools, to assess and translating these electrophysiological or electrical signals into a meaningful behavioral correlates. And then a lot of electrophysiology has been performed to understand the connections within the hippocampus. This is a hippocampus, slice of hippocampus. You have these different cells, they are talking to each other. Depending on each one of these cells, how much information it receives, how long it processes the information, that proportionately translates short-term memories into the long-term memories. You know, someone asked you that, okay, what determines for you to store that information and how long it is going to store? That is based on these connections. So this is all in the hippocampus, and this is the other regions. Okay, there's a lot more complex circuitry. So what we do in the lab is that you take the live slices of the hippocampus from the experimental animals like a rat or the mice, subject these guys to the different disease conditions, and try to understand how the hippocampal circuitry and its physiology is altered in various psychiatric and neurological conditions. So essentially what we do is that, so each one of these nerve cells within the hippocampus, they are talking each other. What is happening is that every time the new information reaching into the brain, its activity increases. And we could make a measure of that, the electrical signals which are generated. So look at this one. So these are the extracellular field potentials which we record from the several neurons, not from a single neuron. The signals are extremely noisy. Can you see here? These are the electrical signals. They appear extremely noisy in nature. So what they indicate that neurons, they are talking to each other, and you stimulate those neurons, you know, they are communicating to each other, the other one, you record its connectivity. That's what we have done. And then what you observe is that these are the electrical signals, and there is whenever the new information reaching into the brain, especially to the hippocampus, even for a brief period of time, and if it is relevant for you, its activity increases. Only those synapses which they are talking together in an activity dependent fashion, its activity increases. And that remains for several minutes, and in some cases it may be several hours as well. So several hours, those two connections are the synapses, they are talking together. So there is an enhanced activity for a long period of time. So we call it as long-term potentiation. So in the hippocampus, how long such enhanced activity is there, how long it is persisting, proportionately that much information what you acquired at that particular point of time translates from short-term memories to the long-term memory. So the activity of the brain which determines the storage of the information or translating the short-term memories to the long-term memories as well. How do you know this? If you block the enhanced activity of these synapses by various means, then you notice that these animals, or even in the humans, they don't remember. They don't translate short-term memories to the long-term memories. The case which I was referring, like HM cases, at various degrees. This is what happens in the aging phenomena, normal aging phenomena. We start losing the connections, and whatever the information which was stored in those button-like structures has been lost over a period of time as a natural process of aging. And it can happen in an accentuated fashion, a disease called as Alzheimer's disease. So you have an advanced aging, so I lose the information. So look at here, the enhanced activity, the block 1 and the red one. The red one is the enhanced activity of the synapses or the neurons. They are talking each other. So it is maintained for a long period of time, something like this. Now, this is what happens. This is a normal one. Before you entered into this hall, your communications in the hippocampus was something like this. So those of you who are very keen to know about how the brain functions, the information, then the synaptic activity will be something like this, a complex processing. And then if you are very keen, then subsequent learning, it recruits the neighboring synapses as well. They are called as the silent synapses. So those of you who are working on, the learning and memory for your projects or the networks, what you will do, you will go and again recapitulate the things. You will try to learn more. So one learning will facilitate the subsequent learning. So it's called as a facilitation of learning happens by recruiting the more connections in the brain, more synapses.
Who triggers this? What triggers this recruitment?
So you have, depending on how long these two are talking together, a specific signaling molecules are released, chemicals which are released.
But you will not do it if it's not relevant to you. So there is some higher portion of the brain which is determining relevancy.
Yes. So in fact, the motivation. So you should be motivated, and also it should be reward based. All our learning happens in a reward based fashion. So without the relevancy, if it is not leading into something good, you are not going to learn.
So probably I have a related question. What it means is there a centralized system on top of this which is directing it, or is it like a distributed system that they talk to each other and?
Okay, it's not a centralized one when you talk about the learning and memory. So what is most important is that, which is not shown here, this is a cell body which is present here, and then you have a process, a dendritic spine, something like this. Okay, so you have the communication between one cell to the other cell which is happening. Depending on how long the activation is there, a particular set of the chemicals which are being activated, the signaling molecules are activated. How long those signaling molecules are activated, depending on that, it may be a local circuitry, it may be local activation, or it could also activate the gene pool. Okay, yeah. So it's something like into the cell body. So it reaches into the cell body, new proteins are synthesized, new signaling molecules are synthesized. They not only activate the activated synapses, they identifying the neighboring one. It's like in a Bangalore city, you have a house, adjacent site is empty. What you will do, you will wait for some time, and then you will just go and plant something, and if nobody is responding, you will put a compound and you will put your name board. Okay. Same thing happens, it's a utility. So it's a use dependent plasticity. Okay, it's completely like that. It's a use dependent plasticity. And it so happens that if you are not using those synapses, it also retrieves back. It's not that all the time you are activating the same synapses. If you are activating the same synapses throughout the day, it's not relevant for you. Only during 1 hour you have a physics lecture, very next lecture is on a mathematics, you have to switch over. So you are not going to recruit the same synapses. So that is the reason probably the nervous system has more connections and more synapses to be activated at any given point of time, very distinctly. Okay, this is what happens. Okay, so don't worry about this pointer. So look at here. So before entering the lecture, at the end of 45 minutes, if you are very attentive, you're supposed to have the alteration in both sides. This neuron and this neuron, both the connections should strengthen. Then subsequently, if it is relevant for you, rather than having 2 connections, now I should have 10 connections. So when I have a 10 connections, my subsequent learning should be facilitated. So how one learning facilitates the subsequent learning? The phenomena is this. This is what happens in the brain. Okay. Imagine that all the time these neurons, the activity is increasing and increasing and increasing. So when does we get back to the normal state? So everything is electrical and chemical communications which is happening. Don't you think so that these neurons, they have to come back to the resting state? That is extremely important. So you have a potentiation and a depotentiation, both should happen. So many years back in 1999, I was looking into this phenomenon. Does the same synapses, you have a switch between both excitation and inhibition. So whenever there is a new information comes, it just shoots up its activity, then it should come back to the normal one, so that the same synapses can be recruited for some other learning, new learning. Does the same thing happens? In fact, in the same neurons, you have both phenomena. So you have an enhanced activity when a new information comes. When the new information and its relevancy and the duration of activation, if it decreases, these guys, they go back to the depotentiation state. Same neuron, same synapse. Same synapses, they have both activation and the inactivation switches. Fantastic, I don't know. Lot more need to be understood. We have not understood those phenomena. Probably this is responsible for the learning and the facilitation, and some cases in accentuated learning. Why some are extremely smart, why others are normals. Normal learning, and some of them are very quick learners. So probably the biophysical properties of these neurons and the electrical activities and the signals which are generated by utilizing the certain set of neurons by some individuals are distinctly different compared to the others, which we don't know yet, which is not completely understood. So we may have to spend a lot of time for that. What is interesting is that biochemically we have the switches. Depending on the amount of a calcium which is present, so which triggers several downstream signaling molecules to activate, which makes both the cells to talk each other. Okay. The calcium level, when it increases, you have the LTP, which I told you. There is an enhanced activity in the synapses. When the calcium level goes down, you have the long-term depression. So you have potentiation and depotentiation, the molecular switch which is very much there in the same synapses or same neurons. It's a complex, it looks a lot more complex. Now, in a disease process, how does the hippocampus react to? I'm not going to give examples of a complex neurological or psychiatric disorders. A very, very simple one. Each one of us, we undergo a stressful situation. So some of the stress, which is always, acute stress is always better. A brief period of stress before your lecture is always helpful for you, because generally your body and the brain arousal mechanisms are triggered. So your blood pressure increases, you sweat a little bit, and the blood circulation to the brain increases, so that your brain is not going to sleep while you are giving lecture. Okay. So this is a general arousal phenomena. It's called as a physiological homeostasis. So every day, every time it happens. But imagine that even though I don't have a lecture, but when I enter into this hall, my blood pressure increases, I start sweating. It should not happen, no. Because I am not giving the lecture, I am only an audience, I will come and sit there. Why should I sweat? Some individuals, it so happens in a context-based fashion, that every time they enter into look at the face of a spouse, or a mother-in-law, or your boss, or your director, or your PhD guide, or your colleague. You have an enhanced response beyond the stimulus. What is relevant in a more irrelevant way, your stress responses are being generated in the brain, because it's anxiogenic. The anxiety levels are very high, which is context based. The moment I enter into this hall, my anxiety level goes up. So that means there was no scope for the brain to habituate. So we get habituated to the faces, we get habituated to the people, we get habituated to the environment in which we live. So it is a natural homeostasis. When that is altered, and every time I am in a stressful situation for a long period of time, and this is what happens. Look at here, these guys. These are the normal cells, the left side one and the right side one. In the chronically, if I stressed out for a long period of time, in the hippocampus, the cells, they lose their dendrites. They are the one which receives the information from the multiple sources. That means I am not able to perceive the information which is coming from the different regions of the brain. Why? Because you don't have these button-like structures. The loss of these button-like structures, they are called as dendritic spines. The synapses are reduced in the hippocampus. And then that translates into an abnormal electrical processing of the information in the hippocampus. So I showed you earlier, every time the new information comes, there should be an enhanced electrical activity. The block 1 is the normal one. So when I am in a stressful situation for several weeks, this is the red one. So if you don't have enough information processing and the electrical signals are not generated in the hippocampus, there is no scope for your brain to translate the learnt information to the memories. It doesn't happen. It's very true in other disease conditions as well. Okay. And also, when you are in a stressful condition, even though on your table you have several things and you bought something new yesterday, but you feel that it's not there. Recognition of the novel objects. When you are in a stressful situation, it will happen to each one of you. You are in a hurry to leave, but you know the car key is very much there in front of your eyes, but unfortunately you are not able to identify it, even though that is not a novel one, even though you have been experiencing the same thing. Why? Because there is no coordination between what you want and what is there in the presence. The presence, the spatial map, and what is your requirement, both of them, they are delinked. Your frontal cortex and hippocampus is delinked at that particular point of time. So that happens when we are in a stressful situation. And also the sociability decreases a lot. Whenever we are in a stressful situation, you don't care about your own close buddies, your own friends. The sociability decreases. So this is beyond hippocampus problem. It's not only the hippocampus, even the prefrontal cortex, which is responsible for you to identify the individuals who are in and around you and their familiarity. That also decreases. And then all of this translates into the formation of new connections and the new neurons in the hippocampus, in the brain. It's only in the hippocampus you have the formation of new cells. Throughout our life, the new cells are added in the hippocampus. That's the only region of the brain you have the formation of new cells. So depending on how much you learn, new cells are added. If you block the formation of new cells, your memories are poor. That's how we know that.
The body's metabolism actually manufactures new cells.
So essentially you have the progenitor cells, that they are the stem cells which are there. So in the other regions of the brain, so once the maturation of the brain circuitry happens, no new cells are formed because that stemness is lost. But only in the hippocampus, because this guy is highly plastic, all the time interacting with the external world, and it's also highly susceptible for various brain disorders. So constantly the new cells need to be added. It happens only in the hippocampus.
In our microprocessors, we don't make new gates. It doesn't make any new gates.
Yeah. So probably if you are planning to design the hippocampus, okay, so you may need to add this dynamicity of the novel environment and the new signaling molecules, because these guys, the new cells, they have a developmental signaling processes. But in the other regions of them, they are not developmental. They are all matured neurons, mature synapses, mature connections. These guys, they don't have synapses, but they do have some of the signaling molecules. The tropic support is provided by these guys. So essentially, the schema which I was talking to you is something like this. So you have the different levels of neural organization. Any perturbation, any disease process could be understood in a better way when we understand at all these levels. Okay. So for the want of time, I will not go into the greater details of the other disease processes, but I will just move into the end of my talk, and then I will summarize a couple of salient things which you may have to think in seriously, so which is relevant for you guys. So you are asking a question: can the remodeling happen in a robust fashion when there is a damage? Can the taking over of functions happen by the neighboring cells or the neighboring regions of the brain? To some extent, yes, it does happen. That depends on the severity of the damage and the degree of damage. So how much brain tissue is damaged? If it is only at the circuitry level and the chemistry of the brain is altered, like in the psychiatric conditions, it's perfectly fine. So you have the neighboring, cells, their neurochemicals which is being released, they try to compensate the loss of other cells or its connectivity by overriding on them. So you do have the reorganization which happens. Now I don't have the cells at all because they have died. In that condition, what happens? So there is a remapping which happens. So if you notice that, here is an example. Here is a digit 3. This is in the monkey experiments this was performed. A representation of different parts of our body, which is very much there in the brain. So left side of our body is represented in the right, and the right side of our body is represented in the left. It's called as a homunculus. Homo means the human, and homunculus means representation, a map. So your body is mapped in the brain. That's how we know exactly if that portion of the brain is activated, I will move only that portions. So you have the maps, which is called as the sensory maps and the motor maps. Look at here the maps. So you have the third digit. The third finger is cut now. When you remove the third one, so look at here, the second one and the fourth one, their territories has been expanded. So there is a taking over of the functions. In absence of the third finger, these two fingers, the neighboring fingers, they work very hard. They expand their territory. So it's a compensatory mechanism which is inbuilt in our brain. It happens. But if the damage is so huge, so imagine that the whole hand is amputated, then this hand cannot be replaced with the other hand. So the scope for the remapping of that one is limited. But adjacent to this hand, you have the face. The face one, it will start expanding its territory. So like a phantom limbs, you might have heard V.S. Ramachandran's experiments. If you scratch his face, you feel that as if his left hand is being, scratched or it is touched. Because misrepresentation of remapping of the body organs happens. So in the absence of one organ, neighboring one, they try to expand it. So even though you don't have the left hand, but if you touch the right cheek, just push it, and then you feel that as if the left hand is being moved.
You don't have any computer program where you can cut out half of it and the other half will compensate for it.
Well, not really. I don't know how you are going to build it. Okay, it's a $1 million question. Okay, for you, how many variables you can add? So say for example, the LTP experiments which I showed you, the potentiation of activity in the hippocampus, as and when we are learning, we are talking somewhere between 500 to 1,000 signaling molecules, known molecules which has been mapped out. So by using a transgenic technology, this technology, you just cut down one by one molecules which are responsible for the activity of the brain. but still there is a persistence which happens. Such kind of a robust plasticity, you do notice in the humans as well. Say for example, you have a stroke patient. A portion of his left brain, a cortex has been damaged. There is no blood circulation. So he lost right hand movements. But the compensation happens to a significant extent by rehabilitative tools. So you are going to drive the right hand, which is non-functional at some point of time, by the electrical stimulations, try to see that whether the connections can be re-established. And if the damage is severe, you lose that completely. It gets completely paralyzed. But what is very important is that don't allow all the cells which are representing this hand in the left hemisphere are lost by the ischemic insult, by the stroke. So as early as possible, if you could restore the blood supply, you are not going to lose, but it is only a partially you lose it. Partial, hemiparalysis, and a partial paralysis happen, it's not a complete loss. So one need to be extremely cautious how quickly you are going to restore it, the neural circuitries back to its function and its metabolism. So using these plasticity mechanisms, one could restore globally the abnormal brain development in the mentally retarded children or the autistic children. The examples, couple of examples which I showed to you is that if you could induce a robust plasticity by altering the abnormal circuitries, translating them into the normal circuitries, where its physiological functions can be re-established, probably most of the symptoms which are associated with a cognitive decline, what we notice in the mentally retarded children or the autistic children, we should be able to restore it to some extent. We are not going to, in any case, we are not going to bring it to the normalcy, but we could facilitate those abnormal synapses to be converted into an active synapses or the functional synapses, which is helpful for the children. That's what we have been doing for several years, and hope we will reach at some point of time. So look at these conditions. You have an abnormal developmental brains, and how are you going to restore it? So the critical period of brain development, and if you use a particular tool, we should be able to restore the abnormal brain circuitries into the normal circuitries to some extent. So this is extremely important. So some children are born with a small brain, something like this, or you can also have a brain something like this. This side is perfectly fine, this side you lost something. You do have cases like this. All the endosulfan victims, if you notice, they have a bigger brains. So in the middle of the brain, you have more CSF, the more water is there sitting, hydrocephalus condition. The brain is pushed into the periphery. That is the reason they don't have the developmental cognitive functions, and also the motor functions are affected. So is it possible for us to help these children who are significantly affected and their cognition has been compromised? It's a social responsibility for each one of us. Before we use all these toxins, how much it affects the brain need to be cautiously looked at it. Many of these changes which happens in the development, the abnormal development of the brain circuitry, which is irreversible in nature. It's irreversible. And most importantly, you cannot transplant the brain like what we are transplanting the heart or kidney or liver. We cannot transplant the whole brain into the one individual to the other individual. Practically impossible, because I have showed you that in the introduction, how dynamic it is, how complex it is, how specific is this connectivity. It cannot be re-established. So these are all the limitations which we have. Hope that by using a comprehensive approach in a multidisciplinary fashion, we should be able to model and also to understand the brain to the maximum extent and in the coming years. And please help us to help the patients. So whatever the tools, whatever the technology you come up, it's perfectly fine with us. Okay. Then it's extremely important. It's a continued rewiring and remodeling of the brain circuits through plasticity mechanisms are responsible for recovery of functions in psychiatric and neurological conditions. So you take a drug therapy, you take a counseling, you take a neurorehabilitative tools, each one of them. What we are doing is we are titrating the plasticity mechanisms, either plus or minus. So in a disease process, there is a regressive plasticity. By using different tools, we are converting this regressive bad plasticity into a good plasticity. Thereby, we could recover the functions. Okay. What is lacking these days is this. You will be surprised why so many individuals are poor in stress management. If the parents are not giving 500 rupees to watch a movie in an INAX, a girl will commit a suicide, jumping from 5th floor or 6th floor. It was never heard. So instinct, it's not a, instinctly, we never do that. Normal humans will not do it. That means the stress coping up mechanisms is absent. Here is an example, look at here. Each one of them are interlinked. Father, mother and a child. Now what we have? Living together, have a child, the child is abandoned. Surrogate mother is taking care of the child. There is evolutionarily, the empathy and the emotions are evolved, and the brain perceives those regions which are responsible for to behave like a humane. That is evolved by the parents and the environment. Now, those who are indulged into antisocial elements, antisocial activities, irrespective of the age, the problem is that amygdala and the neighboring regions are not developed. The good and bad is not taught to these neurons at right age, at right time. When you have an abnormal circuitry, nothing can be done. It is extremely important. You need to have this. Behavior is a function of the brain. Each one of your behaviors is determined by the actions in the brain. Okay. So it's extremely important. You need to have this kind of a social one, which is extremely poor these days. Many of them, they ask, many scientists, they ask, whatever we study in the labs, is it really true? Are you going to translate into the humans and the patient care? Very, very true. Thanks to the nature and for the evolution. So we are all conserved. The evolution has been conserved from lower species to the humans. Nothing is different. That is the reason. But what is most important is that what is the model which you are going to use to answer what specific question of a disease process. That's extremely important. There's a long way for us to go. Having understood the brain is so complex and we are not able to understand many things. We know two things. Brain is responsible for knowing about the external world. And your awareness, you call it as a mind or you call it as a consciousness, whatever it is. The brain and mind, they work together. And they elicit the different states of our mind, these different states of our consciousness. How does this happen? We really don't know. The message for all of you is that there's no difference between Einstein's brain. If I take it and do a biochemistry experiment, the proteins, minerals, whatever you have, everything is there. And there's no difference in my brain. And there's no difference in someone's brain in terms of its chemistry. But what is most important is its physiology, its connectivity and its usage and its functions. If you don't use it, that becomes an archives. If you are using it, that becomes the present. And many of us, we use both, then it becomes a future. So what is expected from each one of you is that you should use the present and also based on the past and develop the future. So that happens if you are consciously using the brain. Thank you very much for your patience here.
I think with this fascinating account, there will be a million questions. So, but we are running short of time. Maybe we have maybe a couple of questions. Then later on, those interested can talk to him offline.
Yeah, please.
One of the slides about the Down syndrome. So, hippocampus is it
Or the Down syndrome, even though genetically you have an abnormality. So genetically you have an abnormal development of a specific circuitries in the brain which is responsible for the cognition. In the Down's patients, what you observe, the Down's children, the cognition is reduced, reaction times are reduced, and sometimes it is hyperactive, sometimes they are hypoactive. Hyperactive is more active and some of them are hypoactive, less active. And also they never associate with the present. So what is happening in front of them, they never associate it. The reason is that I told you that the prefrontal cortex and the hippocampus and the amygdala, all three of these guys, they don't talk in the same tone. There is no sync which happens. That's why whatever the question you ask, whatever the answer is given, there is no relation between both of them. It's extremely important. It's a developmental disorder, but there are certain regimes has been evolved in collaboration with MIT that we should be able to help these Down syndrome children as well by a cognitive therapy, so which is being practiced at NIMHANS, and there is some success stories are there, but it's not all the time 100%. Even if it is 10% success, we feel it's significant.
So genetic disability can be overcome by behavioral
to some extent by the environmental as well, and also the critical period of the brain development is extremely important. How soon you interfere, how soon you advocate the neurocognition tools onto these children, thereby you could restore some of the major symptoms associated with the disease process. It's extremely important, and how soon you are going to intervene. Yes, madam.
Nowadays, I don't know exact term, but some scientists are training students, small children. They call it as a micro-brain training. And in that they close their eyes and try to, so they identify, children are able to identify colors without seeing, and pictures and so on. They join the puzzle, and they are saying that why we are doing this, because they are trying to activate some
essentially,
So is it going to?
No, you see, I will tell you what is required that need to be activated at particular point of time. So you are trying to take the territory of an neighboring cells. So just by closing my eyes, I can identify the colors. So what is the purpose of it? The brain is designed to identify either by touching or by visualizing it. So what you are doing is a abnormal activity. So rather than visualizing a particular color where you have a greater appreciation in a normally, now without visualizing it, I am going to identify something. So you are going to have the remapping studies which I showed you. So you are blocking some other normal functions at the cost of something else. And some children, I see a lot of the signboards in the city, especially JP Nagar, Jayanagar area. I notice some brain kits or something. I don't know what is that. Okay, so it's essentially you need to have a normal environment. I showed you the monkey picture. Have you noticed? Why I'm telling that? It's extremely important. The emotion and the association with the environment and the way we respond and the way we learn, the maturity has to come. The way the child when it is born, when it touches an art plate, it feels that it is hard. The same way you are going to teach each one of them. Now you don't want to accentuate that. Why you want to advance the learning of a specific component at the cost of other things? What is happening is you are compromising the other one. There is no overall development which happens. That is the reason why I gave an example that, so if I don't get 500 rupees to watch a movie in an INOX, I don't jump. If I fail in a semester, so my life is not going to end, because there is no stress management, because it's perfectly fine. This is a bad day which I had and I could not do well. If I don't get a job in this company, perfectly fine. Why can't I take this as a challenge and move ahead and go to the another company and prove myself? based on the earlier bad experience which I had in this company. That is the adaptation, a natural adaptation. So, but the stress management, because in our child psychiatry department, so earlier when we were students, we used to get about, in a day, about 12 to 15 children. They used to come with a different adjustment problems or the behavioral problems. Now it is ranging between somewhere between 250 to 300 in OPD per day. You know what? They're all from the educated pool. Both of them are working husband and wife. The child is abandoned. The mother is a mother always. The surrogate mother or your caretaker is not a mother. The child initially, when you feed, it responds to your hormones. It responds to your pheromones, which is secreted while you feed the baby. That's how the relations are established. If you give in a polyethylene bottle, which is sterilized at 80-degree temperature, it doesn't give that pheromones. So there is no replacement for it. If an emotional development is affected in these children, you have to blame yourself. Don't blame the Cerelac cans or the water bottles which we use, or the milk bottles which we use it. There is a critical period. We have a scientific evidences for that. Up to the child of 5 months, these sensations are developed and the perceptions are developed. Tactile stimulations and sensory cortical developments which happen. And those individuals with a single parent or without mother, with a surrogate mother, for some reason the mother and the child, they could not be together up to 5 months. The sensory cortex which I showed you, that the sensory maturation has not occurred. So it's extremely important. You see the social network, not the present social network, the family social connectivity which was there, grandparents, and then you have the huge families. Now it's all not nuclear family, and if somebody, husband and wife, if they live together with one child, that's called as a big family. Okay, that's the present situation. Okay, so the emotions are built over a period of time, and it is imprinted on the brain. So if I don't care anybody, you know, in evolutionarily we care for each other. No, why you need to care for others? The empathy is a survival. That's a very normal human nature. How many of us we have? We need to question it. You are driving a bike, in front of your eyes some other accident happens. I know that you have to attend this lecture and you don't care about it. 90% of individuals, they don't respond. It's only the 10% of them, they feel that they are part of the society, they need to respond. If somebody is hurt, we need to respond. And you have about 5 to 10% of them, which is a psychiatric illness. If somebody is suffering in front of your eyes and you get more enjoyment than, you know, or you hurt somebody, you get more enjoyment. It's a disease. The amygdala neurons, they seek more. Dil mangte more. They will be keep on firing more. Do something, trouble something. Why are you keeping your colleague happy? Trouble. Okay, all the time, why she has to come first rank? Do something. Snatch away her notes, or trouble, or switch off her, you know, laptop. You see, this is also very natural. That's the same neurons, you know, they function in a different fashion. It does happen. Last question from you. Okay.
There were some slides on meditation.
Yeah.
If you could just summarize.
Now, essentially, now I know as a continuation of that plasticity mechanisms. So, if you want to keep the whole brain healthy, it's not only one region. So, like what you said, that brain kits or something is there. It's only certain regions of the brain you are enhancing its activity. But what is most important is that the whole brain has to be evolved to become a normal human being. So in any case, the current generation, none of us are normal. Okay, let me tell you that. So there is evidences for that. Does we have a global development of our brain? Does we have a global thinking in terms of living in a society? Most of us not. Okay. So the yoga, meditation or the neuro rehabilitation tools, what they do is, it's not activating only one region of the brain, it's the different regions of the brain can be activated. Why that is important? If you look at the schizophrenic patients, different regions of the brain is abnormal, not only one region. Multiple regions of the brain is abnormal. How are we going to solve it? There is no cure for it, let me tell you. So it's only a management. So it doesn't matter, whether you use a yoga, meditation, rehabilitation exercises and going to Tirtha Yatra, it's perfectly fine. But what each one of us we perceive the world, and how many regions of the brain at any given point of can be activated for the good. So till 15 years back, there was no evidence that okay, the yoga and meditation is going to bring about a significant change in the brain circuitry which is going to be helpful. It's not that all yoga is good, all yoga is bad. If somebody who is suffering with epilepsy, if you advocate yes, Sudarshan Kriya Yoga, what Shri Guruji is using it, their seizure frequency will increase. Okay. So they do some jumping yoga, Nityananda Swamiji and all. They're all non-scientific, unfortunate. So many educated people, engineers and everybody are the disciples, they go there. It's very unfortunate, we need to have. So thank God, last 10 years to 15 years, we have a neurobiological basis for different kinds of yogic practices and meditations, has been very, very objectively is studied in great detail, not in India, but rest of the world. We have a wonderful evidences. So we have images, imaging studies, you have electrophysiological studies, you have the biochemical studies by doing the PET MRI. In the whole brain, when you process it and you see that certain chemicals are released when you practice certain yogic practices. And we know that those chemicals are reduced in particular disease conditions. Say for example, in depression, you have certain set of neurotransmitter goes down. If you practice a particular kind of a yogic yoga or a meditation, those chemicals, they come back. There is a natural restoration which happens. So it's a yoga is used as an adjuvant therapy, as a part of our life. That's also like any other thing, like a diet you use it, and you also have some kind of an exercise and are the yogic practices.
So I have just one little tidbit on this to make us everybody laugh. So they have invented garbage collection in the brain long before the computer scientists thought about it. So the yogic practices throw out a lot of the garbage, day-to-day garbage, and in a systematic fashion, which I think would be very interesting for.
Now, in fact, most of our life, if you look at not the current lifestyle, the lifestyle was itself as a yoga. You sit and eat, and you sleep in a particular posture. You write in a different posture, and you walk straight. These are all the postures what you have in the different yogas. So these days, there is no way that we don't do it. So it's close to the Padmasana what you do, when you sit and eat food. So we cannot sit and eat because our, abnormal food diet, your tummies will not help you to bend and eat. Okay, the activity, physical activity is absolutely not there. None of us, largely most of us we don't have. So the brain and mind and the body, all three work together. Body, brain and mind somewhere is there, don't worry about it. So all the three of them, they work. So one is a consciously, many of them unconsciously we work. So until unless you feel that you are within that system, you are not going to appreciate it. None of us we appreciate the world. Always we say that, okay, no, they could have cleaned this. You see, there is a stain here. Why did this management is bad? Okay, but we did that mistake, but we don't correct ourselves. So the same thing happens in our society as well. That is the reason each one of us, our perceptions are different. So the culture is extremely important, and the culture plays a major role. Okay, so we have a scientific basis that how a culture defines your brain and your management of your life.
I think you're
okay. So
course.
Okay, so yeah, thank you very much.